we're going to carry on with a talk from John Walshaw on a core-genome MLST scheme for source tracing of Xylella fastidiosa, which I think comes at a good time after the talks that we have. John is a senior bioinformatician at Fera Science, and he works obviously on Bridget and a range of other projects to provide bioinformatics analysis and support for projects using next generation sequencing technology. So over to John.
So yeah, hello everyone. I'm actually going to give an overview of one aim of Work Package 2, and that's the aim is source tracing of Xylella in the event of an outbreak, and that's included some work from Alan McCluskey and Karen Fraser at SASA, as well as the core-genome MLST scheme of the title, which Sam McGregor and myself have been working on. And before I proceed, I would just like to acknowledge the efforts of everyone involved in this aim, particularly colleagues in the plant protection program at FERA, in bacteriology and the sequencing team. I'd also like to acknowledge Maria Saponari and Lisa Giampetruzzi and Blanca Landa for their interest and some useful discussions. Also at FERA, we've gratefully received some infected plant samples, which I'll say a little bit more about later. So this is about using genomic information at one level or another to compare and characterize Xylella. And just a brief reminder that there are several subspecies of Xylella which have varying degrees of host specificity and indeed geographical specificity. So there's the subspecies, and then they, as we've heard, are characterized in terms of sequence types. So I am just going to give a little overview of MLST to set the scene for some of the later material, which is talking about some finer level typing compared to MLST. So MLST, it involves a small number of housekeeping gene loci, which experts select for each species, and of course they must be present in all of the strains, and they must be well conserved because obviously we have to amplify them and then subject them to high quality sequencing. And there's this well-established scheme with Xylella fastidiosa from Leonard Nunny and colleagues at University of California, Riverside, involving these 7 genes. And there's a range of lengths of the PTR fragments, and of course they're relatively constant in length. And at each of the gene loci, you have one of a number of variants, which we refer to as alleles, and each of those simply has a label. And the way that they're treated is, of course, very simple. They're treated as exact sequences. So to identify one of these alleles, you have to have a perfect match. So 2 alleles which differ by only 1 base pair are treated as different, and that's treated as equally different as if there were many more base pair differences. So it's very conservative and simple treatment. And the numbers we're talking about, I mean, there's sort of a few tens of different alleles for each of these loci. And of course, the unique combination of the alleles which occur at each of the loci is what constitutes the sequence type. So this is just summarizing the first 11 sequence types of Xylella fastidiosa, and there are currently almost 90 of these which are characterized in one particular standard resource, which is the PubMLST database, and there's getting on for 300 different fastidiosa isolates which have got type data in PubMLST. And these data can be used in different ways, but a very simple metric is what I'll refer to as the distance, and that's simply the number of loci at which the core genes differ. So in this simple example, we've got distances of 4 and 1 and 4 loci, and as we've seen, a distance of 1 could be down to a single nucleotide difference. In the context of tracing an outbreak, one way that these can be treated is this sort of representation. If you were to encounter a situation where each of these circles represents a particular strain of a pathogen, and each of these lines represents a distance of just one locus difference. If you were to see this sort of situation, of course, a probable explanation for that is that the outer strains have descended from a founder strain, and of course that's informed also by provenance data, when and where you got the strains, and also the number of different strains that you have observed. So that's indicated by the size of the circles. So you have a founder strain in the middle, very probably, and this is actually data from a real human pathogen. And in this sort of situation, with very many strains which have been recovered and assayed, you may be able to join the dots between multiple founder strains and get a good insight to the spread of the pathogen. Of course, you can't always join the dots quite so neatly. So really, in other situations, it's all about looking at the distances that you have and relative distances, and trying to make sense of those relative differences to give you an indication of what may have happened and where these strains may have arisen. So I just wanted to point out that other kinds of analysis are possible with the MLST sequences. So more sophisticated approaches, the near constant length, lends itself to traditional phylogenetic analysis of either the DNA sequences or indeed the amino acid sequences. And this is called multi-locus sequence analysis, MLSA formally. And I just wanted to flag up that this is something that Alan McCluskey has been doing. And I'm not going to say too much about this, but this just summarizes what he's done with MLST sequences, which he's taken directly from the genome sequence databases. And it does actually illustrate a couple of important points, which are that all of these nodes, these are the different MLST sequence types which are represented in the genomic database. There's not actually that many of them. Also, there's some big gaps between some of the sequence types compared to others. So these are actually nucleotide differences. So some of these sequence types are literally separated by a single nucleotide. With others, they're much further apart. So all of that data arises traditionally from isolates, of course. And again, in the context of an outbreak, we might want to think about streamlining things and weigh up whether we can do things differently. And we've already heard quite a bit about direct sampling from plant material. Can we do MLST like that? And this is also something that Alan and Karen have been doing. So these are actually spiked plant samples. This is fastidiosa subspecies infecting several species of artificially infected by several species of prunus, and the DNA has been extracted by a standard method, and then the normal amplification and sequencing. And the take home is that this has been successful in recovering the sequences, but it does depend on, of course, how much you've spiked them with, and as we've already heard about, different inhibitory factors that you might get from one host to another. So this is a good outcome, which they will be following up with some other methods which they'll be using. So I'm just going to get on to the core genome MLST now. So obviously there's an increasing number of complete genome sequences available in the databases. So very simply, it might make sense to extend the traditional MLST approach just with many more gene loci. So the approach there is to use the core genome, which means the genes common to most known strains. So they may number hundreds, perhaps even thousands. And of course, this should give us the same kind of data conceptually, but at a much finer resolution. But also, and really quite importantly, a very straightforward backward compatibility with the traditional MLST, for which there's a lot of legacy data, perhaps even involved in legislative considerations in some jurisdictions. So I think one of the big assets about MLST is the fact that it's very simple, and core genome MLST is simply related to that legacy data. So in contrast to the normal MLST, where the genes, of course, are curated by experts, we can't do that for an entire genome's worth. And actually even relying on the deposited gene annotations or third-party database curator annotations, we can't really do that either, because there may be inconsistencies, some cases they may be absent. So there's a need to do this automatically. So this sentence is doing quite a lot of work. The equivalent gene loci in each genome are identified computationally. That's glossing over a few issues, which I won't talk about today. And then once you've done that, it's exactly the same principle that you can use combinations of alleles which determine your core genome sequence types. And when you've done that, of course, you've then got core genome sequence types assigned to the genomes that you've used to obtain these sequences in the first place, and of course you can use them to type new ones. And just to say, I'll refer to a few of these. There are software tools around now which aid quite a lot in doing this, but it's not without quite a lot of thought required. So just to define what the core genome actually means, it does not usually mean the genes present in all of the genomes. That's generally impractical. It can be oversensitive. Some genomes are not entirely complete. So there are various definitions using different numbers. It might mean the genes which are present in at least 95% of all genomes or 99% of all genomes. So that also is glossing over a few things. It's not as straightforward as it may seem. It's not completely sort of determined automatically, because this is just illustrating a sort of pathological case. If you have a data set where it's very skewed towards particular, many represent quite closely related, and a few outlier genomes, then you might end up with a situation where all or most of your core genes are all in the same 95% of your genomes and completely missing from the outliers. So you have to impose some other limits, and as soon as you impose some other constraints, then the numbers start adjusting each other in terms of the number of loci, the number of genomes, the percentage that we're talking about, and so you've got a number of interdependent parameters which you need to apply some thought to come up with something that's sensible. But I have no time today to talk about that. So how did this work out for us? Well, we've obtained the available genomes from the public databases. We've ignored lab mutant strains, and for reasons I won't go into, some researchers intentionally do not deposit assembled genomic data, so there's actually quite a lot of other unassembled sets. And so Sam has obtained all of these and he's assembled them, and the short story is we've ended up with more than 200 assembled fastidiosa genomes. And again, glossing over quite a lot of effort that Sam's put in. He's used a dedicated suite of software called Chewbacca, which processes this to produce a core genome scheme. And the result of that is we've ended up with 210 genomes, so small number failed those criteria. We've ended up with a core genome of about 1,400 loci. And by the way, most of these 210 are unique. So there are a few duplicates which are identical in terms of the core genome. And those loci are present in 95% of all genomes, so it's a 95% core, and no genomes lack more than 100 of those genes. That's just an indication of the size of the Xylella genome. And something I'll say no more about is that Sam's also done this independently for the 3 subspecies which are best represented in the databases. So this is also doing a lot of heavy lifting. I'm just going to say data scraping. This genomic information is all very well, but it doesn't mean a lot unless we've got the provenance information, where these things come from. And just to say that quite a lot of work required there, because that's often present but recorded in irregular form. So I've done quite a lot of work trying to, as much as possible, automate the scraping of that data to reduce any sort of future in-house curation efforts that you'd have to do with new genomes which appear. So in terms of what you're actually doing with this data, it's the same concept as before. We've got distances, the number of core genes which differ. So we have a distance matrix between each pair of the genomes. And with any distance matrix, there are lots of mathematical approaches you can take to processing it. All I'm going to say is I've used a not particularly sophisticated method. It's standard clustering, UPGMA, to turn that into a network. And also a bit of work involved in getting this data into this software. There's a variety of software that you can use. I prefer Cytoscape. I think it's very versatile in terms of representing this information. So I've just got a few minutes to give you a quick tour and just highlight a few results. This is what it looks like. And we have lots of fastidiosa dominating here. So the wine-coloured squares are fastidiosa. We've got multiplex up in the northeast and pauca up in the northwest. And you can also colour these by the origin, where they've come from. And no surprise, there's lots of Californian fastidiosa and also some American genomes up in the corner here next to some European samples, strains from multiplex. So, you know, this is a CG-MLST scheme. It's not supposed to represent phylogenetic truth, but we would be disappointed if it wasn't consistent with our knowledge about the interrelationships between these strains and subspecies. So this is just to highlight. So we've got our fastidiosa in the bottom. Say sort of top left is pauca, top right is multiplex, and we see morus in the middle, which fits in with the received wisdom about the origin of that. We've also got the sandii in that location, so that's good. In terms of the distances between them, I mean, it's not really necessary to have a kind of exchange rate between, you know, what does a CG-MLST distance mean in relation to the traditional MLST distances, but we see that there is a significant difference between genomes of the same sequence type compared to those which are different, which ties in with some of those gaps I mentioned earlier. These are the sequence types being shown, of course. So this is just to highlight that this is consistent with some work that's been done showing that various sequence types, not all of them are monophyletic. So Blancalander and colleagues, I think earlier this year, published something about ST6, which there's this long strand here, but it's not monophyletic. There's some more ST6 down here. So it's good that that's also reflected in this scheme. And I'm just going to finish off with a couple of examples. So going back to really what you heard about earlier, some essentially metagenomics sampling directly from infected plants. So this is about a direct sampling of a specimen of naturally infected oleander from Italy, which was independently typed, and it's ST53. And so I'll just sort of whiz through this. This is, standard DNA extraction, but we dedicated a whole MySeq run to this. So we got about 24 million reads. Sam did some metagenomic profiling, and basically it looks like about 5% of the reads were identified as bacteria, but 0.08% identified as Xylella. You can do the maths, see what the probable average coverage is per core locus. So we got about 20,000 reads. Again, cut a bit of a long story short. So Sam assembled this and typed it with Chewbacca, and actually we got 197 core loci where known alleles could be called. So that's less than 15% of the scheme, but it was there and identifiable. And if you plug this into the scheme, it looks good. So this is a part of the network populated by ST53. This is our direct sample. I'm not going to say anything about these are two Salento samples. So, and I just wanted to finish with something we did very recently, which is another type of compromised core genome. So we're actually back to isolates now, but this is a strain which Alan has been sequencing. He was actually trying out a new kit to do long read sequencing, and the short story is he got a closed genome, but it's nanopore sequencing. It's got quite a lot of errors in. About 1 in 7 of the genome looks like it doesn't have errors, and he's going to finish that with Illumina sequencing. But the question is, we just thought this would be a nice test to see if this somewhat messy genome could be typed. And we got 207 loci there. And I honestly didn't know what to call this. So it was X marks the spot. We did actually do this on Monday, and I hope you enjoyed your national holiday. But this also seems to work because this is closest to the actual sequence type ST26, and just got the names of some of the strains to make it a bit clearer. But I've overrun, for which I apologise. So the conclusions, though, are basically we've got proof of principle now, both in terms of the genomes available and the software. The results are in line with established knowledge. We're getting finer detail with the sequence types, and where we've got severely compromised numbers of core genes, we can still get something which looks like it's getting the correct MLST type. And the outputs, we've got our 1.0 scheme, including subspecies schemes, and basically lots of scripts and software which we produced for automation built around existing third-party tools to aid visualization and analysis, and we're preparing manuscripts. And I'd better stop there. So thanks for your attention.
Thank you very much, John. That was really impressive, especially the final bit showing that you could still get meaningful results of a sample like that.